beautiful thing today is that we are going to have our second Ignite talk. And this is a great way to summarize what was happening in nutrient enrichment. They put together a very nice presentation and look at it as a review for the grilling. So, let's do it.
Hi guys, I'm Sarah Lamb.
I'm Ellie Balilos.
And I'm Sayara Fahmy.
And we have the Ignite talk about nutrient enrichment. So let's start off by first defining a few terms. So a macronutrient is an essential element which is used up in large amounts by organisms, whereas a trace element is only required in small amounts, but they can be fatal if there's too much. And lastly, we have our limiting nutrient, which is what we examined in the lab. And this is the element which, in its shortest supply, will limit the population size.
Okay, so Liebig's barrel is a good representation of limiting nutrients. Basically, how it works is the wall of the barrel is made up of sticks, and then the shortest stick, each stick represents a nutrient, and the shortest stick represents a limiting nutrient. So the barrel can only be filled to as high as the shortest stick. Otherwise, the water will start to come out. So this is the shortest stick represents the limiting nutrient.
So a possible question that could come from this chart would ask you to identify the primary and secondary limiting nutrients. So if you look at the population size, when phosphorus is doubled, the population doubles. So phosphorus is the primary limiting nutrient. And the secondary limiting nutrient would then be nitrogen, since the concentration of nitrogen was reduced after the phosphorus input rate was doubled.
All right, next we have primary production. So what is it? It's the conversion from light energy to chemical energy through photosynthesis. And this kind of explains why primary producers are so important in the ecosystem. And as you can see from this pyramid, they produce the most amount of oxygen and energy. Oh, I keep doing that. And this energy is used up in each preceding trophic level. And between each level, about approximately 10% of the energy is lost.
Okay, so one of the other main topics that we covered in this module is competition, which is when both species are competing for a resource. They use the same resources and they both need them. So often it causes a negative outcome for one of the species. There's intraspecific competition, which is when members of the same species compete for a resource. And then there's interspecific, which is when members of different species compete for the same resources. A couple other terms that we talked about were resource partitioning, which is when two species compete, and they're able to coexist by dividing up the resources. We also talked about interference competition, which is when one species can limit the access of another species to a limiting resource. Finally, we talked about exploitation competition, which we defined as the relative efficiency with which the competing species can use up a limiting resource. So generally, in exploitation competition, one species loses over the other.
So I guess now we can talk about how we applied all this information in the lab. So there were two basic experimental setups you could have done. The first was the test tube approach, in which you either examine the effect of a particular nutrient or how nutrients influence the competition between two algal species. And the second approach was the natural approach, which examined the response of algal growth in a natural setting. And in this approach, you used pot plants containing nutrient-blended agar in plastic cups. There were three basic methods for quantifying the algal growth. The first was cell counting using the hemocytometer. And this was significant mainly because it was the only method that's able to distinguish between two species, and you use this method to examine competition. The second was the optical density using the spectrophotometer, and the last was chlorophyll A concentration, which took advantage of the fact that all photosynthetic organisms need chlorophyll A concentration, but you still can't distinguish between two different algal species.
All right, and then just a reminder, we've already gone through the t-test and the rank sum test as statistical tests we use, but in this module, we're introduced to the Spearman rank test, the Spearman rank correlation test, excuse me, and this examines the correlation, if there's a correlation between algal growth and the concentration of some nutrient. So an advantage of this test is that if it is statistically significant, you can definitely say there's a correlation. However, if it's not statistically significant, your results are inconclusive. And I think we're out of time. So thank you. I hope that summed things up.
Any questions?